It's a real honor and a privilege to be here talking to you, and I must say every time I come to this conference I find it very educational and moving and motivational to me and to people in my lab as well to hear all of the real stories from those of you who really have to deal with these diseases, and it does a tremendous amount to stoke our fires in the research world to solve some of the problems that are ahead of you and all of us together. What I'd like to do today is give you a brief introduction into some generalities about prion diseases and then move on to update you all on our recent progress in, I guess I've got to figure out how this works, and how to diagnose prion diseases in living individuals. And so, just to remind you all of what you probably already know is that all of these prion diseases are also called transmissible spongiform encephalopathies, are slow, fatal, and as yet untreatable and transmissible neurodegenerative diseases that occur in a wide variety of mammals, including cattle, deer, elk, and moose in terms of chronic wasting disease, the prototypical prion disease that we've known about for hundreds of years, scrapie in sheep, then of course the human forms of prion disease. And in humans, these diseases can come in either sporadic, familial or acquired forms, the sporadic being the most common one being sporadic CJD, which occurs in about 1 person per million people per year around the world and accounts for something like 90 to 95% of human TSE cases. And these are not associated with any known mutations in the person's prion protein gene, and we think this probably amounts to spontaneous disease in the individual that occurs and then spreads to the point where it causes disease. A smaller subset of these cases are familial. They involve mutations in the person's own prion protein gene that are pathological and ultimately can lead to various prion diseases, including familial CJD, GSS, and fatal familial insomnia, to name some of them. And then a still smaller, much smaller subset of these diseases are what we'd say acquired. That is, they were initiated by some sort of external infection, such as Kuru in New Guinea, iatrogenic CJD, iatrogenic meaning from medical mistakes, such as inadvertent inoculation of the infectious agent, the prion, into people via, say, neurosurgeries, dura corneal transplant, growth hormone injections, things like that, a tragic occurrence. And then, of course, variant CJD, which we all assume came from people consuming BSE or BSE-infected cattle. So the infectious agent of these diseases, I mean, not all of, in fact, only a very small subset of people acquire the disease, as I mentioned, due to infections, but in almost all cases, the disease they have is in turn transmissible to others, maybe not easily, but it's transmissible. And the infectious agent that is responsible for this represents a strange new class of pathogen, in contrast to, say, viruses or bacteria or protozoans, in that there's little immune response by the host to the pathogen. The pathogen has no genes of its own that it carries around from host to host. And it's also legendary for being hard to decontaminate, especially by the conventional means of disinfection that are used in hospitals and clinical settings and so forth. And it's become clear that these infectious agents actually represent an infectious form of prion protein. And here's an electron micrograph of some purified material, highly infectious prion protein in the form of what we call an amyloid fibril here. And this stuff is, I don't know if I'm blocking your view for some of you, but this is very, very small. It's 100,000 times thinner than this line here. A particle like this would contain hundreds of prion protein molecules, and these particles can grow at the ends and in turn also bundle together into much larger structures that accumulate, say, in the brain in the form of what we call an amyloid plaque. And yeah, like I said, it can accumulate in brain in various forms, not just amyloid plaques, but other forms as well in brain and a number of other tissues as well. So how is it that this type of pathogen can propagate as an infectious protein? Well, it turns out then that it is an abnormal form of a host protein. So all of us, and all of our, if not all of our tissues, express a normal form of what's called the prion protein, which functions as separate units, as shown here. And this normal prion protein we often call PRPC for the cellular form. I know the terminology is very confusing here, but we've got prions as the infectious agent that can be made of prion protein, but it's made of an abnormal form, an infectious disease-associated form of prion protein, I don't know why that happened, that we call, a lot of us call PRPD for the disease-associated form, or PRPSE for PRP scrapie. And this form differs from the normal form by having undergone a shape change and clumping together into these particles that, that contain several to hundreds, if not thousands, of misshapen prion protein molecules. And so how does it propagate in a way that could allow it to be infectious and to spread throughout individuals and between individuals? If we look just at a host cell here, a generic host cell here with the nucleus with its chromosomes and its genes, there's a normal host gene that encodes the normal prion protein, or can be abnormal if it's mutated. But in this case, all you need to do is have the normal form that's encoding the normal form of prion protein. And if one of these abnormal infectious clusters comes along and interacts with that cell or enters that cell, it has the ability to grab onto these normal forms and convert them to more of the abnormal form. And this process can go on and on and on until you make large amounts of these fibrils grow, and you make large amounts of them and end up with many more. In fact, you can put many more infectious particles than you started with. In fact, you can put a single infectious unit into a person or an animal and end up with 1 to 100 billion infectious units in the clinical stage of disease. So propagate these agents really do. So not only do they have to be able to propagate in the sense of making more of the material, but they also have to spread within individuals and to some extent between individuals. And so within people, the spreading routes include, things are instigated either spontaneously, as I mentioned, in the case of sporadic CJD, or can be instigated by medical procedures that inoculate a little bit of this infectious material into the brain usually, or they can be started by ingestion of infectious particles. But from wherever that initial site of misfolding occurs or introduction of the infectious prion, it's necessary for the prions to be able to propagate and spread into the nervous system if the infection is initiated peripherally, and then through the nervous system to the point where they build up to high enough levels to cause disease. And so this spreading then and accumulation in the nervous system is really necessary for disease. But having said that, I'd like to emphasize that human prion diseases, at least, are although they're transmissible, they're not typically contagious in any casual sense between individuals, at least nowhere near to the extent that they can be contagious, such as chronic wasting disease in deer and elk, which really does seem to pass from one individual to another fairly efficiently. So for some reason, and we don't understand all the reasons why, there must be weak links in this overall human prion transmission cycle, such that the prions composed of this PRPD are not efficiently shed into the environment from an infected person, or not efficiently taken up in such a way that they can really instigate a productive and clinically relevant infection. So, and maybe the biggest difference, I'm not quite sure, between humans and, say, deer and elk is simply we have much better hygiene that prevents us from taking up large amounts of stuff that other people might have shed into the environment. But that remains to be really determined. So with that introduction, I'd like to turn to then this issue of how can we better detect and diagnose prion diseases as early as possible in people while they're still alive, and hopefully when, or maybe even before they're sick in the case of familial TSE diseases. And I think, as you know better than anyone, there is a real need for definitive diagnostic tests for these diseases as you get clear answers as early as possible, and as treatments come along, you can begin treatments as early as possible. And not only do we need to be able to diagnose diseases that are already occurring, but we also like to have very, rapid, very sensitive and rapid tests for the presence of prions, so we can prevent infections in the first place that might be due to contamination of a wide variety of materials that become are of concern from time to time. And so the current diagnoses of sporadic CJD, and I'm no diagnostician or clinician, I have to say I'm a biochemist, so forgive me if I'm a little wrong on some of this stuff. But in living patients, we're sort of restricted to coming up with a probable diagnosis of CJD based on a variety of clinical signs and other types of tests that can be done, none of which in themselves are definitive enough to say, oh yeah, this is for sure CJD. So the diagnosis of definite CJD requires a demonstration of the abnormal PRP, the PRPD or PRP-CJD in nervous tissue. And usually that requires then either a biopsy or an autopsy post-mortem, which of course is not the optimal situation by any means. So we and a lot of other groups are trying to develop better diagnostic tests based on detecting the abnormal prion protein in living patients. So that and there's quite a number of tests being developed. I know you can't read this. In fact, I can't even read it from here. But this is a long list of the type of tests that people are developing that are really ultra-sensitive and or have varying levels of sensitivity anyway, and have various advantages and disadvantages. And they're all based on detecting the abnormal form of prion protein. So, but at the end of the day, I think we're going to need multiple tests. Not so that we can have primary diagnostic tests, but not only do you want a primary test, you want confirmatory tests, probably based on a little bit different approach, so that you're as sure as possible of the validity of that test to improve the specificity of the test and minimize false positives. In other words, of course, it's terrible to tell somebody you've got a positive test for CJD when they don't really have it. So we really want to minimize that possibility as much as possible. And we're going to need to cope with a wide range of sample types and applications with these tests. So it's really good that we're having a lot of labs developing different types of tests that will be hopefully complementary and reinforce one another. The type of test that we've been pursuing most in our group is largely the work of these two at least initially, these two postdocs in the lab, Jason Willem and Christina Oru, and also Ryuichiro Atarashi, who was a postdoc in my lab and now is at Nagasaki University, was really the founder of most of this technology in my group, and we've worked together on developing this sort of approach. And you end up with a test that he called real-time quaking-induced conversion, or RT-QUIC. And basically, it's a fairly practical way of detecting prion-seeded PRP amyloid. And how it works is you have a little plate that's about this big that's full of 96 holes in it, or wells. And you, into each well of that plate, you can put your test sample that may come from some human tissue, along with a vast excess of the normal form of prion protein that we've made in bacteria, and a stain that lights up brightly when it interacts with amyloid. And so if you have a prion in your test sample, or prions, shown here, it can grab onto the vast excess of the normal prion protein we put into that tube, basically, and cause it to make these fibrillar amyloid structures that then can bind this dye and fluoresce. And so when you put this plate into the instrument that measures this fluorescence and shakes the plate and incubates it, what you see over the course of the time of the reaction is if you have prions in your sample, you'll see a sudden, oftentimes if you have a lot of prions in there, a sudden increase in fluorescence that this instrument can read. And in fact, this is really incredibly sensitive because you can take up to 1 billion, that's with a B, billion-fold dilution of, say, brain tissue from a scrapie-infected mouse or hamster, and put it into the reaction and still get positive responses, even 10 billion-fold sometimes. So these tests are extremely sensitive, amplifying the presence of the prion by billions or even trillion-fold, depending on the permutation. It can be quantitative. You can tell the relative amounts of these prion particles in samples with these tests, and we've designed them to be incredibly disease-specific in a sense that if we put, say, an Alzheimer's brain sample in there, or Parkinson's or Huntington's or whatever, we won't get a positive response. It's specific for CJD. And we think, so far, are much faster and cheaper than similarly sensitive tests, but that situation is changing every time you turn around. So we and a lot of other labs have developed a variety of applications of these sort of tests to detect all the different human forms of prion diseases, but also a bunch of different animal prion diseases. And most of this development's been done using brain samples, but of course that's not optimal to have to rely on brain samples for diagnosing these diseases in people or animals. So a lot of effort's been made to apply these assays to much more accessible diagnostic specimens, such as cerebral spinal fluid that you get from spinal taps, nasal fluids or brushings, and I'll tell you about that more in a minute, blood samples, saliva, urine, so forth. So a lot of labs have contributed to the expansion of the application of these assays in much more practical ways. So I just want to highlight a couple of these applications, and in particular, to start out with, applications of this RT-QUIC test to cerebral spinal fluid as a diagnostic test for human CJD. And focusing on a couple of major studies that have been published by Ryo Atarashi and his colleagues in Japan and Australia. In this paper here, and another study by Alison Green, Bob Will, Richard Knight, and postdoc Lynn McGuire in the UK, with a little bit of coaching from us. And these studies together showed that this approach can have a pretty high diagnostic sensitivity of 77 to 89%, usually at the higher end of this range, And what we mean by sensitivity is the percent of CJD cases, or the cerebral spinal fluid from those cases, giving positive responses in our reaction. With 99 to 100% specificity, which is the percent of non-CJD samples that give negative responses. So this is really good performance, and represents the first disease-specific diagnostic test that doesn't require either a brain biopsy or post-mortem analysis. So it's really significant work here. And this approach is being implemented by many CJD diagnostic centers around the world as a real definitive test in living people. And so now, as nice as this is, we were a little worried about the fact that still between 23 and 11% of the cases that might be, of the CJD cases that might be coming in the door were being missed by this test. So you'd want to catch all of them if you could, not only to provide real decisive information to the patient and the doctors, but also to, because of the possibility that the disease could be transmitted to other people through hospital settings or neurosurgeries or whatever. So we set out to improve the conditions of this test and ended up with a new set of test conditions that improved quite a lot on the older ones by making them faster and stronger responses than we were getting before. So this then are average tracings in the RT-QuIC reaction as a function of time, using either the new conditions that we have here or the old conditions here. And what I think you can see, even if you don't understand what the graph's really representing, is that we're getting a much faster and stronger reaction using these new conditions, using cerebral spinal fluid samples from 48 different, this is an average from 48 different patients here, than we were getting with the old conditions. And we can also pick out a larger percentage of the cases than was possible before. So we're reducing the time required for these tests from a matter of, say, 2 to 5 days now to a matter of hours, which is really important practically. And we're now getting a higher sensitivity too, so far 96% sensitivity and 100% specificity of this reaction, which is better performance than the old conditions. And it's not only my lab that's been getting these results, but also Jerry Safar's lab and another lab as well. So this seems to be reproducible in a variety of settings. And so these new conditions then improve the performance and practicality of these definitive ante-mortem diagnostic tests. So another approach that we've taken, though, so that we don't always have to rely on spinal taps or cerebral spinal fluid samples, is to try to get samples from the nasal cavity. And one thing about the nasal cavity, if you look at this side view of the brain, you can see that the brain comes in very close proximity to these sensory cells, the sensory neurons that line your nasal cavity, right up at the top of your nasal cavity, gives you your sense of smell. They're the detector cells for smell. And they're right across this very thin bone layer. In fact, they penetrate right from the linings of your nasal vault into the brain, as you can see here through this little bony layer. So this sort of gives a direct window to the brain, more direct than any other anatomical site. So what Gianluigi Zanuso did, who's an Italian neurologist who actually recognized that there's abnormal prion protein lining the nasal vault here some years ago, he said, and when he heard about our RT-QuIC test, he said, well, maybe I can figure out a way to sample this tissue very simply with a little brush and send it to you in Montana for testing with your assay. And he did that, and you know, maybe we could come up with a really good test. So he did that. He devised this method where you just go in and take a very gentle brushing of the nasal vault, and you drop it into some liquid and send it to Montana, and we've done the test, and apparently it's not very painful at all. A student who has worked for both of our groups underwent this procedure as part of an instructional video, and she says, you know, it's just like having a little, it's not comfortable. It feels like you have a little extra water up in there that shouldn't be there or something, but it's not overtly painful, she said, and this is what people seem to say. I know some nasal brushings can be painful, but this one doesn't seem to be, from what we've heard. So it turns out that this is working really well. Again, this is another comparison of the kind of response we get from nasal brushings. We call these samples OMs, for olfactory mucosa samples. But anyway, these nasal samples, you can see, give much stronger, again, and faster reactions than we get from the cerebral spinal fluid samples from the same patients. These are using the old conditions. And we also pick up a higher percentage, much higher percentage of the cases using their nasal brushings than we do their cerebral spinal fluid samples. So we're getting, in our initial study, we're getting positive reactions from 42 out of 43 CJD cases, but not from 43 non-CJD patients. So basically we were getting 97% diagnostic sensitivity and 100% specificity, relative to 79% sensitivity using the cerebral spinal fluid or spinal tap samples from those same patients. And so we think then that these nasal brushings provide a potential basis for a definitive and less invasive antemortem diagnostic test for CJD. At the same time, something that's a little more disturbing was when we use this assay to measure how much prion seeding activity is in these nasal brushings, we found that each brushing contained between 100,000 and 10 million seeding units, which means there might be substantial infectivity lining the nasal cavity. But the neurologist we talked to in John Luigi, Dr. Zanuso, is saying that it's very unlikely for that to come out, and most of it will, it's so high in the nasal vault that most of that, if not all, would go down the other direction and into the gut and not come out into the environment. But it's something we have to think about, even if it's probably not overtly a huge deal. We're not quite sure. So lastly, I just want to say that we've found now, I mean, one of the problems with this sort of test is that you need a lot of different types of normal prion protein substrates, different ones to add into these reactions to test for different prions that come from humans and animals. In fact, there's quite a few human prion diseases, especially some familial forms, that were undetectable by our test and other tests as well. And so we've recently found, though, that we can use a prion protein that sequence using the genes that we've isolated from a little animal called a bank vole and express it in bacteria that serves for some reason to be a universal sensor or substrate for this RT quick reaction. So we can, using this single protein, it's much more practical now because we can use this single protein it's a single reaction and detect all the prions that we know about in mammals, coming from, you know, wide variety of animal species, and also all the different prions that we know about in humans that we've been able to test so far, including, as I mentioned, these ones in red, ones that have never been detectable before with these sorts of in vitro assays. And the sensitivity is often comparable to what we were getting with other substrates. So we think this is improving the practicality. And the other thing we can do now, using this particular substrate as a sensor molecule, as we call it, along with other sensor molecules, we can actually discriminate, say, between sporadic and variant CJD with this test on the cerebral spinal fluid and presumably nasal brushings as it comes along. But because with the one substrate we pick up, whoops, I don't know why that happens. We can pick up both sporadic and variant CJD very, very sensitively, whereas with another substrate shown here, we would only pick up sporadic CJD. So in one fell swoop, on one plate, we can discriminate between the two, which is a nice practical advantage. So to conclude then, we think these RT-QuIC assays are becoming increasingly practical and sensitive and specific. This bank bowl prion protein seems to be a universal sensor molecule for the RT-QuIC assays, again making it more practical, and helps us perform strain discrimination based on the relative detection with different sensor molecules and biochemical comparisons of the products of these reactions. In our fantasy, and this is the last slide going forward, is to envision the possibility that similar types of tests can be developed for all these other protein misfolding diseases that involve, at least to some extent, amyloid accumulation, such as Alzheimer's, Huntington's, Parkinson's, ALS, lots and lots of important diseases that can be confused with CJD. Similar tests might be developed, and that patients with early neurological signs could, as soon as they come into the clinic, could be tested right away with a battery of these sort of tests to say, okay, these are the proteins that are misfolding in your, especially from nasal brushing, say, in your body, and so this is the disease you really have without having to bounce around between a whole bunch of different clinicians and centers over long periods of time. Get a definitive answer to what your problems are. And then, because this definitive answer becomes early, it makes it much more likely that therapeutics, the treatments will be effective, because instead of having to reverse a whole bunch of damage that's already done because it's taking you so long to get the answer, you can say, nip it right in the bud while people are still, pretty good shape. And so it makes therapeutics a lot easier. I mean, there are, as time goes on, therapeutics will come along. The other really nice thing about all this is that in attempting to develop new treatments, you can use these sort of tests, especially in conjunction with a non-invasive, not so painful nasal brushing samples. You can do multiple samplings over time in patients that are being given the treatment or not given the treatment, and do clinical trials and get a more rapid answer to whether the treatment you're attempting to develop is effective or not, by looking at whether the pathological protein that is causing the disease is being eliminated or at least prevented from propagating itself. So with that, I just want to thank a bunch of people. I have a fantastic group in Montana who's continuing to do remarkable work, and previous postdocs as well. I mentioned these people before. Got a number of different laboratories who are, and this is really a crucial part of it, including Dr. Safar, who are providing, giving us clinical samples to test and to develop our work with. And I also want to thank especially the CJD Foundation and donations from some of you, specifically from Zoe and Jenny Smith, and their mother, to help this work move forward. So appreciate your attention.